Good day. My name is Shreyal Maikoo, and I am a PhD candidate at the HIV Pathogenesis Programme based at the Nelson R. Mandela School of Medicine in Durban, South Africa. Today I will be presenting some of my PhD work titled The Effect of HIV-1 Subtype C Transmitted Founder Virus's 5' LTR Genetic Variation on Viral Reservoir Size and Latency Reversal Potential. Even though combination antiretroviral therapy, or CART, as a treatment for HIV infection has significantly decreased AIDS-related deaths over the years, we all know that CART is not curative. This is due to a subset of latently infected cells harboring transcriptionally silent integrated proviral DNA, which is rapidly established following infection and is referred to as the HIV latent reservoir. Due to this block in transcription, no new virions are being produced by these latently infected cells. Therefore, these latent cells are resistant to clearance by the host immune system and are also not recognized and targeted by antiretroviral drugs. The persistence of this viral reservoir continues even during long-term CART and also explains the rapid rebound in plasma viremia soon after treatment interruption. Therefore, lifelong treatment is required. Given the persistence of the stable latent HIV-1 reservoir, many strategies have been put forward to either eradicate or permanently lock the latent virus, and these are termed shock and kill and block and lock respectively. I will be focusing on the shock and kill strategy, which focuses on reactivating the latent reservoir to induce viral replication and then subjects the infected cells to the host immune system and viral cytopathic effects, thereby depleting the infected cells that constitute the latent reservoir. So the shock and kill strategy makes use of latency reversing agents, or LRAs. There are several different LRAs or different classes of LRAs, each acting via different pathways to reactivate the latent virus. This previous study showed that when resting CD4 T cells from infected individuals on suppressive antiretroviral therapy were treated with different LRAs, it caused statistically significant increases in intracellular HIV-1 mRNA levels. However, these different classes of LRAs do not uniformly activate all the patient latent viruses to the same extent. So this brings us to the question of what causes this differential reactivation. So this then made us look at the HIV genome. Firstly, before we look at the genome itself, it is important to note that HIV type 1 exhibits a wide genetic diversity of subtypes and recombinant forms, with subtype C being the predominant subtype in Southern Africa and is responsible for 50% of infections worldwide. So particularly within the HIV genome, one of the genes that our group focuses on is the long terminal repeat, or LTR, which is the promoter that drives viral gene expression and replication. There is also the TAT gene, which encodes a transactivator of transcription, or TAT protein, which transactivates the viral promoter and enhances transcription elongation. So it is unknown what the effect of the LTR genetic variation is on the observed heterogeneity in the patient responses to latency reversal that I just showed in the previous slide. Furthermore, as I've highlighted previously, latency is established very early in infection. Approximately 80% of heterosexual transmission events and infections are established from a single HIV-1 variant, and this is called the transmitted founder virus. Our group's recently published data showed that when we looked at the patient-derived LTR's response to latency-reversing agents such as PMA, TNF-alpha, prostatin, and SAHA, we can see here that there is a variation in the reactivation by these latency-reversing agents. And we also saw a similar variation when we included the consensus TAT. However, the effect of the transmitted founder virus's LTR genetic variation on viral latency development or reversal is unknown. Hence, the study seeks to address this. So this brings us to the aim of my study, which is to investigate the effect of the HIV-1 subtype C transmitted founder viruses LTR genetic variation on the propensity for latency establishment and reactivation potential by latency reversing agents. And we hypothesize that the HIV-1 subtype C transmitted founder viruses LTR genetic variation may modulate the propensity for latency development and reversal. So my study is broken up into 3 goals. However, for the purposes of this presentation, I will only be focusing on goals 1 and 2. So my goals are also further divided into several objectives. So the first goal is the construction of HIV-1 subtype C transmitted founder LTR pseudotype viruses and determination of their propensity on latency development and reversal in Jurkat cell lines. So the objectives here was firstly to construct the consensus HIV-1 subtype C LTR and TAT pseudotype virus expressing a GFP reporter gene, and then the establishment of the latent T cell lines harboring this consensus reporter virus. Thirdly, it was the construction of the patient-derived HIV-1 subtype C LTR pseudotype viruses expressing a GFP reporter gene and the establishment of the latent T cell lines harboring this patient-derived virus. And lastly, the comparison of the patient-derived LTR pseudotype viruses for their propensity of latency establishment and reversal. So this is the methodology that was used to achieve the first goal, where we developed a cell line-based model of latency for subtype C, prior to which we did not have any model for subtype C. So we termed this model the JLAT-C model. Prior to this, already in existence was the JLAT model, so J for Jurkat cell line and LAT for latency. So this was a Jurkat cell line-based model of latency. So basically what we did was customize this model for subtype C. So we did this by cloning in gene blocks for consensus HIV-1 subtype C LTR and TAT into the P-blue script expression vector and then into the PEV731 HIV minimal reporter genome plasmid. Usually, this plasmid contains the subtype B LTR and TAT, but what we did was then cloning the subtype C LTR and TAT with the GFP gene, and this formed the minimal reporter virus for subtype C, which we called the C731CC. This was then co-transfected into the 293 T cells along with the VSVG and the R8.91 plasmid containing all the other viral proteins to generate the fully functional virus containing the consensus subtype C, LTR, and TAT. This lentiviral vector was then used to infect the Jurkat cells to develop the JLAT-C model, which contains the latent virus, and GFP expression was then measured. And we then compared the propensity of HIV-1 subtype B and C to develop latency and their reactivation potential by latency reversing agents. So our results firstly show the successful development of the JLAT-C model. So in figure A, we can see an outline of this process where on day 0, we infected the Jurkat cells with the C731CC virus following measurement of productive infection on day 4 post-infection using flow cytometry. So we measured this productive infection by the percentage of GFP positive cells in panel 5. And we also infected the cells with different dilutions of the virus, such that approximately 5% of the cells would be infected. So this would mean that the remaining 95% of GFP negative cells in panel 4 are not expressing GFP, meaning they are either truly negative or they are the latent cells. So when we reactivate the cells, if they do reactivate, then that means that they were latent. So these are the cells that are then sorted for the GFP negative population. And we then reactivated these GFP negative cells after determining the optimal dose of PMA, which resulted in the highest reactivation of cells with the least toxicity. So following reactivation on day 5, we were then able to get as much as 0.5% GFP positive reactivated cells. So then in figure B, we show that cells harboring the latent virus express GFP with subtype B showing to be more reactivatable than subtype C. So after these observations, we then suspected that maybe TAT isn't efficiently expressed in subtype C compared to subtype B. So the Western blot then shows that actually TAT was equally expressed in both the subtypes. So this suggests that maybe subtype C has a higher propensity for establishing a more latent provirus than subtype B. However, further experiments will be conducted here to measure the amount of integrated DNA copies of the virus to ensure that it is the same between subtype B and C minimal genome reporter virus, so that we can then check whether subtype C is forming a more stable reservoir compared to subtype B. So the HIV-1 subtype C patient-derived LTR has been previously cloned into the PGL3 basic vector in our lab. So I have then amplified the patient-derived LTR and cloned it into the C731cc plasmid. And I then sequenced the patient-derived LTRs just to ensure that I still have the same sequence that I initially amplified. So then on the right-hand side is a phylogenetic tree where I compared the LTR sequences that I have cloned into the C731cc with the initial sequences that were cloned into the PGL3 from which I amplified them. And I was expecting each patient ID to cluster together with equal branch lengths since they should have been the same sequence. Unfortunately, I was able to see that with these samples. Suggesting that the cloning was successful. We then went on to produce the virus with these patient-derived LTRs and infect the Jurkat cells and measure reactivation potential using different latency reversing agents indicated at the bottom left of each panel. So for example, this was PMA, TNF-alpha, prostatin, and SAHA, as well as different combinations. And our data demonstrate that all 20 patient viruses exhibited a differential reactivation when stimulated with the different LRAs alone or in combination. And this observation is similar to our previous findings in our recently published paper that the patient-derived HIV-1C transmitted founder LTR genetic variation exhibited a differential response to the extracellular stimulation with LRAs. So this then brings us to goal 2 of my study, which was the determination of HIV-1 subtype C transmitted founder LTR pseudotype viruses, propensity on latency reversal in primary CD4 T cells. So the objectives here was to firstly establish an ex vivo primary CD4 T cell infection latency model system and perform an in vitro latency reversal assay in primary CD4 T cells. And secondly, to compare the propensity of patient-derived pseudotype viruses expressing GFP to establish latency in primary CD4 T cells by evaluating the response to different classes of latency reversing agents. So following the observation that there is differential reactivation among the patient-derived LTR pseudotype viruses in the Jurkat cells when stimulated with different LRAs individually or in combination, we then postulated whether we would see a similar response in the primary cells. To this effect, we then isolated CD4 T cells from 6 healthy donors and infected these cells with 6 of the 20 patient viruses. We just chose 2 highly reactivatable viruses, 2 moderately reactivatable viruses, and 2 less reactivatable pseudotype viruses. So our data demonstrate that all 6 patient-derived HIV-1C transmitted founder LTR pseudotype viruses reactivated significantly above the threshold dotted line following the addition of the LRAs compared to the unstimulated control, which are the black bars in the primary CD4 T cells obtained from all 6 healthy donors. And furthermore, once again, there was a differential reactivation among the patient-derived pseudotype viruses when stimulated with different LRAs in all 6 healthy donor primary CD4 T cells. So our data once again suggests that the HIV-1 subtype C transmitted founder LTR genetic variation may be a contributing factor to the heterogeneous response to LRAs. I would like to acknowledge the following individuals and institutions. I thank you for your time and I welcome any questions. Thank you.